Today we'll be talking about gel electrophoresis. By the end of this video, you should be able to describe the use of DNA electrophoresis to analyze DNA fragments, determine the percentage of agarose needed to resolve a given size of DNA, define the components needed for DNA gel electrophoresis, describe the electrophoretic migration of uncut plasmids, and predict the migration of DNA fragments. Previously, we talked about how to isolate plasmid DNA and determine its concentration and purity using a spectrophotometer. However, that's not going to tell us everything we might want to know about the DNA, like its size, for instance how long or short it is, or its shape, whether it's linear or whether it's circular. So we're going to use a different technique called gel electrophoresis. Gel electrophoresis relies on two properties. First, that DNA has a net negative charge, which is going to allow us to move it with a current. And second, that we can use a matrix of agarose to discriminate the DNA based on its size. Shown here is an image of an actual agarose gel matrix. You can see that the agarose fibers form pores. When we add DNA to the gel and apply current, the DNA will be forced through the pores. Small DNA will move more easily through the pores, while larger DNA will migrate more slowly. So gel electrophoresis allows us to separate DNA based on size. Additionally, we can control the density of agarose in our gel. Typically, we might use a 1% agarose gel, which would give us a broad resolution range. However, if we had several small pieces of DNA we wanted to discriminate, we could increase the density of agarose in our gel. This would decrease the size of the pores and thus separate small-sized DNA from each other. Conversely, we can also reduce the density of agarose, which would allow us to tell the difference between larger pieces of DNA. Thus, the density of agarose in our gel determines the resolution of different sizes of DNA. Here's a cartoon of a typical agarose gel setup. The gel is in the center of a tank that is filled with electrophoresis buffer. The gel has wells, which are where we're going to load our samples or our DNA ladder. A DNA ladder is a set of linear DNA molecules of known size. By comparing our samples to the ladder, we can estimate the size of our DNA. Before adding samples to our wells, we first add loading dye to the sample. Loading dye has two important components, glycerol and a dye. Glycerol causes our sample to sink in the well and prevents them from diffusing out of the well. Dyes are used to track the migration of our samples through the gel. We'll be using bromophenol blue, which migrates equivalently to a 300 base pair linear DNA molecule. Here's a picture of an actual gel setup. You can see the agarose gel here in the middle, the wells with and without loading dye, and the tank, which is filled with electrophoresis buffer. The electrodes are here on each end, and you can remember that DNA always runs towards the red electrode. Runs towards red. When we're done running our gel, we're going to remove the gel from the buffer tank and stain it with an intercalating nucleic acid stain called gel red. Gel red binds specifically to double-stranded DNA and allows us to visualize the DNA under a UV lamp. We can also take a picture of our gel. You can see here the wells, and then the DNA ran this way through the gel, with the larger pieces being closer to the wells and the smaller pieces being further down through the gel. This is an example of a DNA ladder. We know the size of each one of these bands, and because of that we can estimate the size of our sample in our other wells. Everything we've talked about so far is true for linear DNA. However, DNA can also have more complicated shapes. That's important because the rate of migration is dependent on both the size and the shape of the DNA. Plasmid DNA exists in three forms: as linear DNA, as a relaxed circle, or as supercoiled DNA. Supercoiled DNA is DNA that has been twisted to reduce how much space it occupies. This is important because there is a lot of DNA in a cell. For example, the DNA in a single human cell would be longer than 6 feet if placed end to end. Plasmid DNA also can be a relaxed circle. This happens when one strand of DNA breaks, which is also referred to as being nicked. The nicked DNA can then relax from the supercoiled state into circular DNA. If the DNA has a break in both strands, it becomes a linear DNA molecule. Importantly, even though our plasmid is the same size, it will migrate very differently depending on what shape it has. Since supercoiled DNA is compact, it's going to migrate the fastest through the gel. The other two forms of DNA will run similarly, with the relaxed circular form running more slowly than the linear form. Importantly, linear forms of DNA are the only ones we're going to be able to estimate accurately, since their migration is not complicated by shape. So let's practice. We have a hypothetical 4 kilobase pair plasmid, and we want to run it on a gel. What would we expect to see in this case? Well, we would see all 3 forms of DNA. We'd have a supercoiled form that would run faster than the linear form or the relaxed circle. Now, if we take the same plasmid and digest it with a restriction enzyme, we'll see something a little bit different. So instead of all 3 forms, it'll collapse down into a single band. That's because the enzyme is going to cut the supercoiled and the relaxed circular forms and convert them to the linear form. By linearizing the plasmid, we'll be able to accurately judge the size of the plasmid based on our DNA ladder. Now instead, if we cut the plasmid with 2 restriction enzymes, we're going to see something a little different. In this case, we'll have separated our DNA into 2 pieces, one that's 3 kilobases long and one that's 1 kilobase long, and we'll be able to distinguish these 2 forms on our gel. So now you should be able to describe the use of DNA electrophoresis to analyze DNA fragments, determine the percentage of agarose needed to resolve a given size of DNA, define the components needed for DNA gel electrophoresis, describe the electrophoretic migration of uncut plasmids, and to predict the migration of DNA fragments. Thanks for listening.